Sri Gurubhyo Namaha. Welcome to our online learning initiative, and I welcome you all for the biology class. Last class, I gave you a question and I told you to solve it, right? So come, let us solve together. Yeah, here is the question. So, haplodiploid sex determination system as a special characteristic feature. So, it is a haplodiploid. So, what is haplodiploid? Haploid is nothing but N, okay? And diploid is nothing but 2N condition. 2N is seen in female and N is seen in male. From this only we are going to solve. Okay. So one is over. And then next, as a special characteristic features such as the males produce sperm by mitosis. So mostly that is gametes totally they will divide by what? Meiosis process. But here it is about mitosis process. So another clue is here and it totally they are given the answer here. We have to justify the answer. Okay. So they are talking about a male drawn. They do not have father, number one, and thus cannot have sons. Justified. Then they have a grandfather and can have grandson. So, two things we have to justify. So, let us have a recap about this honeybee. The honeybee, female honeybee, queen bee, only will lay egg. It lay two types of egg. One is that is it going to fuse. Another one they do not fuse. Okay, I told you the term, the process also parthenogenesis. So one is which is going to fuse. They all will become into a female. The one egg which is not going to fuse with the sperm will develop into a male. Okay, so this point is very clear for us. And now they're talking about your grandson, grandfather. So we have to take two generations. So whenever you're reading the question, okay, stick towards the point. Okay, what point they are asking? Here they're asking two things. One son, another one about the son and grandfather. So two generation we are going to take. So let us start. First generation, the female with XX chromosome and male with one single X chromosome. Pull out the gametes, X, X, and here only one. It is a haploid condition. We discussed in the beginning itself, right? So these are all the gametes, and this is parent. And now this X chromosome, when it fuses with the male X chromosome, it becomes XX. Okay, we are talking about honeybee. That sex determination in honeybee is different from human. Okay, so children don't confuse. Observe now. So this is XX chromosome. This XX chromosome can develop into two types. Okay, one, it is well nourished. Okay, that particular thing will called as queen bee. When it is poorly nourished, it becomes worker bee, and all these worker bee will be sterile. Okay, and the queen bee fertile. And here, if you see, this X chromosome. Okay, don't go for this. You will do a mistake here. You'll go for this X chromosome. We are talking about the queen bee's egg only. In this egg, okay, I will use another color so that we can differentiate. So this X chromosome, okay, will divide mitotically, okay, and then they become a drone. That is male honeybee, right? So this is generation 1 we are talking about. Now this male honeybee in generation 2, okay, I'll use the same color. In generation 2, okay, when it happens to intercross with a female with haploid or diploid, it's a diploid female. And then I'll use the same color, X chromosome. Okay, now this X chromosome will fuse with the X chromosome of what? Female, right? X chromosome, and they become into what? female. Again, this female will be of two condition. Either it will become a queen bee if it is well nourished, or it will become a worker bee, right? Another X chromosome here. Okay, I will encircle this with another color. Another X chromosome of queen bee, okay, will undergo mitotic division and they become male, right? And this is drawn. Generation 2 here. We are talking about only male. See, we are talking about only male here. Okay, now we'll read the question and we'll verify that. So, see here, they do not have father. Okay, we'll take this one, generation 1. In generation 1 itself, we can say whether they have father. Grandfather only you have to go to 2 generations. So, we'll take in first generation itself. So, here, check here, it is a female egg. That egg only it's keep on dividing further and further and develops into a male. So, they do not have father. Verified. Then next, they thus cannot have sons. So check out this. Okay, in second generation, happen to fertilize with a female and check whether do they have son. When this X chromosome fuse with the another X chromosome of female, it becomes a female honeybee only. Only the egg of the queen bee, when it keep on dividing without fusion of the sperm or the drawn X chromosome, it will only develop into a male. Okay, so here this is also verified. They cannot have sons. So again we'll verify. They do not have father. Yeah, they do not have father. Thus cannot have sons. We'll check in second generation. When this drawn going to fuse, okay, the fused one is developing into a female only. There are no sons. So verified. Next, they have a grandfather and can have grandson. So now we have to go for 2 generation. We'll take this fellow, right? Here, okay, this drawn will take it. So this second generation, go back, okay, we want to say that they have grandfather. So this is a female. Whose female we are taking? This female will take it. And go back, okay, and then check here, grandfather, okay, so this is mother, okay, grandfather, here is the grandfather, got it. So this is also verified. And now they have grandson. Check this grandfather now. Go back and this grandfather's sperm fuses with the egg here. Okay, then and then from here it is fertilizing. Okay, and so here is a grandson. Yes, so hence it is verified. They have grandfather, they can have grandson, but no father, no son. A very simple answer it is, right? We'll move to the next one. The next about theories, we saw Mendel's law, 3 laws we saw. Okay, law of dominance, law of segregation, law of independent assortment. And now we are going to see about chromosomal theory. Two different scientists, Sutton and Bower, okay, they both proposed this theory, okay, and then they were checking with Mendel's concept, okay, then they gave this inheritance theory. So we'll see now. Chromosomal theory of inheritance. When they have clubbed with the Mendel's work, they are given the term as chromosomal theory of inheritance. It's a 1 mark question, children. Make a note. Who proposed the chromosomal theory of inheritance? Sutton and Boveri. Okay. And the year 1902, you can see. What they said that the pairing and separation of a pair of chromosome during meiosis lead to segregation of pair of factors. Okay, a very simple. Whenever we are learning about chromosome, okay, think about that division, chromosomal division, when it happens during cell division, how the chromosomes are pulled apart. Okay, if you see, 1 minute. If you see, the chromosomes are paired up, okay, and how they are pulled apart, segregation, okay. The homologous chromosome during mitotic cell division and meiotic cell division, you can see it is pulled apart on either side with the help of spindle fibers. Yes, that concept you have to keep in mind here. What we read in 11th standard, the type of cell division, 2 types of cell division, mitosis and meiosis, right? The homologous chromosome, how it pulled apart. Now we'll read and see. It said that pairing and separation of pair of chromosome. I'll draw here the pair of chromosome. This is the chromosome. Okay. This is pairing. Okay. How the homologous chromosomes are paired. And segregation, how it is going to pull it apart. Just recap, we saw about prophase, metaphase, anaphase, and telophase. All right, you have to think about the stage. Our work, we are not going to write a stage here. We have to know that how they are going to pull it apart. Okay, in which stage, right? And then during meiosis, mostly we'll concentrate here meiosis. Okay, because gamete formation, right, lead to segregation of pair of factor. Factor again. Okay, don't think it's a new term. It's nothing but gene. Okay, next slide I'm going to show that parallelism. Okay, parallel, both are parallel. What the chromosomes are doing, the same thing seen in the gene also. Okay, how these both are parallel with each other in their working mechanism, that we are going to see. Okay, yeah. Chromosomes, before going into the parallelism, we'll see about chromosome. Okay, we'll just refresh about the chromosome. Chromosomes are vehicle of heredity because it carry the gene, so it's acting like a vehicle. Two identical chromosome form homologous pair. Okay, I have drawn that. Once again I'll draw, just check it out. Two. Okay, and this you call it as homologous pair, right? Gametes are present in a linear fashion on the chromosome. So if this is the chromosome, the gametes, if you see the gametes, a linear, okay, in a line, the gametes are present in a line, a linear fashion. Okay, the genes also we can say. Yeah, they segregate at the time of gamete formation. Okay, that is, they're talking about the cell division, mitotic cell division, meiotic cell division. Independent pairs segregate independently of each other. When they are talking about independent pair in meiosis, okay, that is, they will be in a bivalent condition like this. This is pair A, okay, pair A, and this is pair B, okay. So independent, they are not dependent on each other. Independent pairs segregate independently of each other, will be segregated. They will not depend on A, will not depend on B, B will not depend on A. Okay, that's what they are trying to say there. Then chromosomes are mutable. Yeah, mutation can happen anywhere. Okay, suppose if it's happening in the gene, means where is the gene? The gene is there in the chromosome only. So naturally chromosome will also mutate. Okay, sex chromosome determine the sex of an individual. So in human, if you take it, XX chromosome, XY chromosome. So if it is XY, it is male. If it is XX chromosome, it is female. Yeah. So this is a structure of chromosome. It's looking like X-like structure, right? And now we'll see the parallelism of the gene. So we have some knowledge about the chromosome. We know about the gene. So far we read about the Mendel's law. Let us compare and contrast if anything is there. This is chromatic, what I'm showing here. And this is the DNA. Okay. Then next, the chromatid will condense and they become into a chromosome. Okay, when? During cell division. And this DNA, inside the DNA, the genes are there, the codons are there. They keep on coding for a amino acid in the protein or form, right? So we are going to see the parallelism. A Mendelian factor as well as chromosomes are transferred from generation to generation, both. Okay, first point. They occur in homologous pair. Which one? Homologous pair chromosome. Yeah, I'll draw the structure so it will be clear for you. The genes also occur in pair. Gene means you don't go for the point that locus all. Just to take a well-known example, our capital T, capital T. It's nothing but the genotype, right? They also occur in pair. Then next, both chromosomes and genes segregate at the time of gamete formation. Yeah, this one during mitotic cell division or meiotic cell division. Whatever the cell division, they segregate. And this TT also like a gamete. we pulled out, right? I'll write once again. Capital T, like this. They segregate. Yeah. Different pairs of chromosomes segregate independently of each other. Similarly, one pair of alleles segregate independently of another pair. This one, different pair of chromosome segregate independently of each other. That I've shown already. Okay. Once again, I'll draw with different color. Yeah. So different pair. I told in previous slide, A, P homologous. Why they are segregating, they will not depend on each other. The same way, okay, in dihybrid cross we saw capital T, another one capital Y. I've taken size of the plant, color of the cotyledon. They segregate independently and they come as capital T, capital Y, capital T, capital Y. Yeah. So hence it is verified this point also. Fusion of two gametes. Let me erase this so you can see the point very clearly. Yeah, fusion of two gametes brings about diploid chromosome number as well as the allelic pair in the offspring. Okay, two gametes means they are talking about X from father, okay, X from mother. So the offspring is called as female, XX. Okay, so it is a diploid. X will be having only haploid set of chromosome. Okay, if it is a male, from male, if you are getting the chromosome. Okay, if it is a male, yeah, one X, another one female. Okay, that is also haploid. The fused one in the zygote, if you see, it will be in diploid. That's what they are saying here, diploid chromosome. As well as allelic pair in the offspring. Allelic pair, how they pair up? Okay, after that, okay, like I'll write again. Capital Y, we saw there, no? Capital T, capital Y, capital T, capital Y. Two gametes, when they fuse, they become as what? Capital T, capital T, capital Y, capital Y. Right? So, we saw about the parallelism. And next, we are going to see about the other scientist, Hunt Morgan. So far, we discussed Mendel work with Pisum sativum. Okay, then Sutton and Bovary's concept in about chromosome. Okay, what was their theory? And now we are going to see another scientist, okay, who conducted experiment in a fly, fruit fly. The fruit fly, otherwise called as Drosophila melanogaster. I'll show you the picture. You can see he is Hunt Morgan. Okay, he selected, let me show you. Yeah, this is the Drosophila or fruit fly. You can see two varieties here. Okay, one, by seeing the color, you can identify white color, mutant type due to mutation, and red eye, white type. When you say it is a normal type, it is okay. Red eye Drosophila, dominant also you can say, and the female and male can be differentiated by their abdomen. Yeah. So we'll see the characteristic feature, like why he has to select this Drosophila. There are more number of insects and animals. Why you want to stick with the Drosophila? Okay. So it breeds quickly. It has short lifespan. See again, like Mendel selected pea plant for the short lifespan. Okay, it breeds quickly. It has more contrasting character. The same thing here. Breeding can be done throughout the year. Okay, because of the short lifespan, and because of that so many progenies can be generated and you can get the result very easily. They can grow on a simple synthetic medium. You need not have like a honeycomb hive valve. Okay, in a simple medium it can grow. Male and female flies are easily distinguishable by their abdomen. We call this as sexual dimorphism also. Okay, the distinguish between male and the female. Yeah. So we have seen about Hunt Morgan, okay, then about Sutton and Bovary's concept. Next class, we'll see at another concept. Thank you.